Okay, dear students, today we are going to study about the Mendel's law, and being based on that Mendel's law, the first law we are going to study. It is called the first law of dominance, and Mendel has illustrated his first law experimentally by performing his experiment on the pea plant in the following way. And today here I am going to show you how the monohybrid cross, that is the cross that is performed by involving out of the 7 pairs of contrasting character that Mendel has studied on the pea plant, among any one of the characters, today we are going to perform the monohybrid cross. So it is like this, you may see here on the board. So let's see monohybrid cross. So in order to perform or in order to study the monohybrid cross here today, I am going to show you in a chart here, and I am going to take, and out of the 7 pairs of contrasting character, I am going to take the height of the pea plant, and according to Mendel he has performed the monohybrid cross in this way. Let's see here. So in order to perform this monohybrid cross, first of all we need to take the parents. Here we'll take the parents of pure characters, pure parents, that is pure tall, pure tall, and pure. This is the parents, or we can simply say parent. Similarly, then after genotype, pure tall genotype means, since according to Mendel tall is the dominant character, that's why the genotype of tall is represented by the capital T. And since we have already discussed in the previous class, that is any gene or any allele is always represented in a pair, that's why T and capital T represents the genotype of pure tall. Similarly, pure dwarf, since it is the recessive character, it is represented by the small letter of the dominant trait. Then after, there is gamete formation. Gamete formation, it forms the 2 types of gametes, T and another is also. In this way it forms the gamete. Similarly, its gamete are also formed in the same way, small t, small t. In this way the gametes are formed. After this we'll perform the cross fertilization to obtain the offsprings of F1 generation. Mendel has performed the cross fertilization between the pure parents of contrasting characters. So when we perform the cross in this way, capital T, small t, T small t, capital T small t, and capital T small t. Now this result belongs to, this is the result of F1 generation, where it has given the 100% hybrid tall, that is all the offsprings that were formed after the cross fertilization between the pure breedings, that is between the contrasting characters of the pure organisms in the F1 generation. That means all the offsprings formed in the F1 generation were found to be tall. That means here they are called the hybrid tall, since they contain the gene to the genotype of the hybrids contain the genes of let's say they contain the heterozygous kind of gene, that is capital T and small t. So although small t represents the pure dwarf and capital T represents the pure tall, since capital T representing the pure tall is the dominant characters, all the offsprings developed in the F1 generation expressed to be tall, although genotypically they contain the gene of the dwarf also, but while expressing, since tall is the dominant character, capital T is the dominant character, it suppress the recessive character and express itself. That's why all the offsprings that are formed in the F1 generation, when we observe them phenotypically or when we observe their morphological characters, then they appear to be tall. Now after this, after the formation of the F1 generation, the same offsprings which are formed in the F1 generation becomes the parent for F2 generation. Now next, parent for generation, its phenotype will be hybrid tall. Then genotype, its genotype will be capital T, small t. Then gamete, gamete formation takes place. Now while forming the gamete, they form the two different kinds of gametes, that is capital T and small t. Now while performing or while obtaining the offsprings of F1 generation, here Mendel has performed the cross pollination or cross fertilization, but while performing the or while obtaining the offsprings of the F2 generation, here Mendel has performed the self pollination or selfing. Here to obtain the F2 generation offsprings, we have to do the selfing. Now while doing selfing, let's do selfing here. Selfing or self pollination. Then selfing or self pollination means the cross with itself or others like itself. That means while performing the self pollination, will perform the cross between the two different organisms having similar kind of gene in this way. Or let's do it in a table. Let's perform the selfing in the table. In this way we'll make a table. This represents the male, this represents the female, and the male gamete capital T small t, and the female gamete also, since we are performing the self pollination or selfing, will make a cross between the two opposite organisms having similar kind of genes. Now let's make a cross between this T and this T. It gives capital T, again cross between this T and this T gives capital T small t. cross between small t and capital T gives capital T small t, and cross between these two gives small t small t. So this is F2 generation of spring. Now here we can obtain the result of F2 generation. Suppose here I am going to give the result of F2 generation. In F2 generation, here we have obtained, let's see the result of F2 generation over here. So in F2 generation we found three different kind of genes. This TT we found, where TT represents pure tall, capital T small t represents hybrid tall, and small t represents pure dwarf. Now if we consider the percentage wise, or out of total 100% of offsprings, according to the Mendel result he has obtained in the second generation, the pure tall he obtained the 25% result, hybrid tall he obtained the 50% result, and pure dwarf he obtained the 25% result. So, and after this we have to write down the ratio, phenotypic and genotypic ratio. Now sorry, therefore phenotype ratio, phenotype means it is the external morphological characteristics. Then whenever we look or observe the results of F2 generation, phenotypically we can study two different characteristics in the offsprings produced. For example, out of the 100%, or let's say out of the four organisms formed in the F2 generation, here we can see this capital T small t appears morphologically tall, this capital T small t appears morphologically tall, since it, although it contains the gene of the recessive characters, but since the capital T is the dominant character over here, the T small t gets suppressed and capital T gets expressed. That's why phenotypically or morphologically the organism having the gene capital T and small t appears to be tall. So if so, this one, first one, capital T, it appears tall. This also appears morphologically tall. This also appears morphologically tall, and this appears capital T, sorry, small t, it appears morphologically dwarf. That's why phenotypically, while making the cross between the hybrid organism here in F2 generation, we found that, or we can obtain the result that out of the four offsprings formed in the F2 generation, three are found to be tall, they express tall, and one organism is found to be dwarf. That's why phenotypic ratio, we can find the ratio of tall is to dwarf, that is the phenotypic ratio of monohybrid cross will be tall is to dwarf, that is 3:1, where tall here will be percentage wise. If we express tall percentage will be 75% and dwarf percent will be 25%. So this is about the phenotypic ratio. And next is the genotypic ratio. Let's see here. Genotype ratio. Now genotype ratio, this result belongs to the genotype ratio. In genotype ratio, or while finding the genotype ratio, we will observe the gene. For example, the offsprings produced in the F1, F2 generation with this kind of gene, that is capital T, capital T. Out of 4 organisms, the organisms having this kind of pure tall gene is found to be only 1, that's why 1. And this kind of gene producing organisms in F2 generation are found to be 2 here, so capital T small t are found to be 2, that's why 2. And small t kind of gene containing organism in F2 generation is found to be 1, that's why the ratio is 1. That's why the genotype ratio of monohybrid cross is 1:2:1, and this is the ratio of 1 means pure tall, is to 2 means hybrids, and this one means pure dwarf. And here percentage wise, pure tall means 25%, hybrids means 50%, and pure dwarf means 25% in the gen. So in monohybrid cross, the genotypic ratio of monohybrid cross for every organism. Now here we have taken only one kind of contrasting character since it is a monohybrid cross, and in case of monohybrid cross, in whatever the characters we perform, our result or our genotypic ratio is always constant. That is, genotypic ratio of monohybrid cross is always 1:2:1, and phenotypic ratio of monohybrid cross is always 3:1. Since already we have discussed in the previous lesson, phenotype is the physical or morphological external appearance of an organism which we can study very easily, and genotypic is the genetic makeup or genetic constitution, or is a kind of characters which we cannot observe since it is based upon the gene. And looking at this chart, according to the Mendel's experiment, we can come to the conclusion that the organism in the context of genotypic ratio, here 1 represents the pure tall, since here in this chart we can find the organism with pure tall kind of gene is only 1, and 2 represents the hybrid tall, because since this is the hybrid and this is also the hybrid, that's why altogether there are 2 hybrids, that's why 2 in the ratio. And organism containing the pure dwarf kind of gene is present only 1, that's why 1:2:1 is the genotypic ratio, and 3:1 is the phenotypic ratio of monohybrid cross. So in this way we can perform monohybrid cross in any of the 7 pairs of contrasting characters studied by Mendel on the pea plant. Now, being based on the Mendel's experiment, other biologists such as T.H. Morgan, before also I have already told you, this Mendel, he was given the title of the father of genetics only after his death, because whenever he has proposed this kind of theory, his findings were published on the local journal of Austria. That's why other biologists from the outer world were unknown about his findings. And later on, almost 40 years later, other biologists from the Germany like T.H. Morgan, Tischermark and Hugo de Vries from the Holland or the Netherlands, Dutch biologist, Dutch botanist. They studied about the same characteristics, and T.H. Morgan, Thomas Hunt Morgan, has performed the same experiment of Mendel in the another organism, that is Drosophila or the fruit fly, and whatever the result he has obtained in while making the cross between the Drosophila having long wings and short wings, the result obtained by him were totally matched with whatever the result obtained by Mendel. So that the other scientists supported, other biologists supported Mendel's, and Mendel was honored as a title of father of genetics only after his death. So in this way we can perform the monohybrid cross in the pea plant among the various kind of 7 pairs of contrasting characters. So this is all about the monohybrid cross. Okay, thank you.